Houston, we've had a problem here.
13 Minutes to the Moon, Season 2.
I was thinking that we weren't going to survive.
Episode 1, available now.
Hello, you're listening to Crowd Science from the BBC World Service. This is the show that takes your questions to the scientists that might have answers. And so we're here at the University of Oxford's Nuffield Department of Medicine to meet the researchers who are tackling the issue of the moment and the science behind it because of this question from listener Evelyn.
Hi, Crowd Science. I'm Evelyn, and I'm calling from Westminster, Colorado in the United States. I'd like to know what scientists are researching to help us combat the COVID-19 disease. In particular, I've heard that some groups of people, such as children, could be infected without showing symptoms and could unknowingly be carriers. Is this the case? If so, how can researchers and health officials address this?
Thanks, Evelyn, and everyone who's sent us questions on this topic. Producer Jen and I have gained access to some of the key laboratories working against coronavirus. I'm grateful to the scientists who've let us in, given how busy they are right now. Coronavirus has changed both the focus and the minutiae of their working lives. Just listen to us nervously negotiating a handshake.
Hello, nice to meet you. Are you still shaking hands, or are we? Oh, that's reassuring.
Washing them a lot, though.
Yeah.
Peter Horby is Professor of Emerging Infectious Diseases at the Centre for Tropical Medicine at the University of Oxford. He's one of a whole team of researchers working around the clock to find out more about COVID-19 disease.
It's a difficult area, epidemic infections, because you have to move very quickly because the outbreaks are pretty quick. We've been working initially with colleagues in China since 2nd of January, and it's been very full on. 7 days a week, often calls at midnight or later.
So what are you working on right now?
So we're doing a few things. We've been trying to collect standardised clinical data because a lot of the data that comes in is partial and it's quite biased. So what you hear about is the sickest patients in hospital, but what you don't see is all the patients in the community, all the people in the community who've got a cough and sniffles and get better. And so you end up usually with a rather dramatic picture of this very serious disease with lots of deaths, and you really need to understand the full spectrum of illness because there'll be a bunch of people who get infected and don't get sick at all. There'll be a bigger bunch of people who get infected and get a mild illness, and then the top of this iceberg are the people who get into hospital. So we've been trying to collect data to give us an idea of the full iceberg, including the bit you can't see under the water. So we've been working with colleagues around the world and with World Health Organization on trying to collect that clinical data.
It feels like that might be key to why this novel coronavirus is spreading so much, is that there's a proportion of people who suffer with it who don't actually technically suffer at all.
It's clear that it's very transmissible, like a lot of respiratory viruses, and that a lot of people don't get very sick, and so they're not really aware they have infection, and it's passed on like that. There may be some transmission even before symptoms start, but it's likely that people are most infectious at the time that they start to feel symptoms.
Our listener Evelyn is interested in people who display milder or no symptoms at all, particularly children. Are there patients like that in the data?
It's becoming clearer that children do get infected, but they seem to generally either have no illness or a very mild illness. There are some children who've got quite sick, but the risk is quite low in that group, so that's good news. It's not yet clear how they contribute to transmission of infection. So in flu, children are really important. They really drive transmission of flu, and so things like closing schools, etc., can have a big impact. With this coronavirus, it seems that that's probably less the case, that children do get infected, but because they're not very symptomatic, they're likely to be less infectious, and so they play less of a role in spreading infection.
Symptomatic, meaning coughs and sneezes that transmit the virus on?
Yes, so the more symptomatic you are, the more infectious you're likely to be.
Evelyn's question is right on the money then, particularly with COVID-19. This strain of coronavirus can infect some people without presenting any symptoms for days. If we trust the data, around 30% could have almost no symptoms at all, but others will be hospitalised. This presents a challenge for researchers. How on earth do you find the people with coronavirus whose bodies are giving little to no clue that they're harbouring it?
And that's where different types of studies become useful. So taking blood from the general population and looking for antibodies, which you get after infection, is really useful, because that's the only way you get a full picture of how many people were infected. And then you get the idea of that full iceberg of how many people were infected.
Epidemiology is not the only thing Peter and his colleagues are studying. So we're going to come back to him later. But as he just mentioned, antibodies are another part of a different vital response to COVID-19 disease, which is happening inside everyone who's infected, our immune response. Time to find an immunologist.
I'm Tao Dong. I'm
Nice
Professor of Immunology.
Bumping elbows with me is Professor Tao Dong. She's Director of the Chinese Academy of Medical Sciences, Oxford Institute. Originally from China, how did she end up working in Oxford?
Ooh. That was very, very long time ago. 28 years ago, I came here with my husband. And then I started my DPhil study in Oxford.
And now, has life here changed in the past couple of months because of the coronavirus outbreak?
Yes, very much. To be honest, I didn't sleep very well since it started, because I come from China. So we get very updated information from colleagues and friends, and at all levels. Some are really professionals and world-leading experts in China, and some just our friend, family who have no knowledge about immunology or science, just like grandparents, uncles, aunties. Everybody is worried.
And they're all texting you?
Oh, this is your field, I think. Because this is a completely new virus, new thing, I think everybody is worried and panicking.
And what are you working on right now?
So I'm an immunologist. I'm also Oxford director of this new institute. This institute will prioritize the funding to support projects, including my own project. For example, in terms of immunology, we study the immune response in patients who is doing well and not doing well. And this is largely in collaboration with our colleagues in China, Beijing Yuan Hospital and the Chinese Academy of Medical Science, because they have collected patient samples from different outcomes with different outcomes, which enable us to study the immune response properly.
Can I ask how you collaborate with colleagues in China if China's on lockdown? I mean, do you send samples through the post?
This is a long-term collaboration. We started our collaboration in 2002, so we've been working together on different viruses and different outbreaks like HIV, SARS time, and avian flu. One of the reasons we want to have this joint institute is we can react very fast when something like this happened, and it's not just matter for the locally, it's a global problem, and it's need a global effort to really join force and combine the strengths, sharing the resources, reagent and the knowledge and expertise to fight as quick as we can.
Tao's team are already set up to fight events just like the COVID-19 disease outbreak. As humans increasingly encroach on animals' territory, they're unwittingly meeting millions of tiny microbial organisms living inside those animals, some of which could infect us. So scientists figured this disease, or something like it, has been coming for a while. One thing Tao mentioned there a moment ago were reagents and the importance of sharing them. What's a reagent?
This is a really fantastic work Chinese scientists have done. Is they published the sequence, virus sequence, very, very quickly. That means we can just, by looking at the sequence, we can synthesize. So now, just by having that sequence, we can make the protein in 2 weeks.
What is the protein? You're not building the virus again?
Spike protein, just because that's important immunogen for making antibodies for vaccines. So spike protein is very immunogenic, and this is it's outside of the virus, so it's quite important.
I should say to our listeners, Tao is kind of making her hands into a ball in the air. I feel like you're trying to describe the virus if it were a ball. Can you sort of give me a description of what coronavirus looks like?
You have like a ball inside, then you have all this, what is called? It's very hard to decide. I'm just thinking about. Okay, so in my daughter's school, some boys have this very gelled hair, spiking, spiking hair. It's a bit like that, singing star, It just points to like different directions, but it's quite distinct. So that's why you want to make a kind of antibody. They can bind to it, then they can stop it.
You're listening to Crowd Science from the BBC World Service. It's time for us to head into the lab where scientists will be unpicking the virus to work out how it gets into our bodies and cells and how it interacts with our immune system.
Don't touch anything, okay?
We won't.
We're here answering listener Evelyn from Colorado's question, what can scientists do to combat COVID-19 disease?
Word after finish.
Okay.
Okay, let's go here.
Coronavirus isn't the only virus Tao and her team are working on. It turns out that from a scientific perspective, there are some similarities between coronavirus and another well-known virus, HIV.
We learn a lot from HIV immunology study, and lots of techniques we are developed is actually from HIV research. For example, Isabella is a world expert in one technique. It's called flow cytometry.
Hello, my name is Dr. Isabella Pedrosa. I am a senior scientist working at the NDMRB. We focus on vaccine development, especially in the context of HIV infection.
So the experiment Isabella is very good at is one of the key experiments we want to do with the patient samples. Hopefully she will show you with the machine and how the machine works.
This place was built 6 years ago and appears to be everything you'd hope for in a lab tackling some of the world's most challenging diseases. White benches, computers, glass walls, all very high-tech. As we head towards a separate room where Isabella's kit lives, a blue-gloved scientist dashes towards the door.
Sorry, we'll be like two minutes.
Science gets priority, and this machine is in high demand. Once the files are transferred, Isabella guides us in to see the flow cytometer.
This is a flow cytometer. As you can see, this is a machine that is around 1 meter times 1 meter. The flow cytometer in immunology plays a very, very important role in research.
This is a machine that allows them to look at the physical and chemical properties of cells. The team will use this to try and work out why two patients with the same coronavirus can react in very different ways.
We can tease out the differences in the immune system of each individual and compare it between populations as well, and we can analyze millions of cells at once. So the flow cytometer plays a very critical role in terms of vaccination strategies.
So we're in the processing of getting some samples here, so it will happen.
Okay. So currently this machine is being used for other viruses, but as soon as you've got the blood samples...
No, we're probably not going to use this machine. We have to have a very safe environment which is approved, so it will happen in a Category 3, Schedule 5 room designated for COVID-19.
Category 3 means I'd have to put lab coats and face masks on.
Yeah.
Time to leave the lab. Fire a good scrub, just in case. Back in Tao's office, I wanted to know how COVID-19 patients' immune systems are responding to the disease. Research into this topic is really early days, but from what's already out in the literature, Tao's particularly interested in a reaction seen in some patients called a cytokine storm. To fight off an infection, our immune cells need to coordinate their attack. They do this using a set of proteins as chemical messengers. These proteins, called cytokines, give the immune cells their instructions. They're also responsible for the reactions, fever, aches, runny nose that you know of as symptoms of many colds and flus. Now, occasionally, severe infections set up this disastrous positive feedback loop, producing way too many cytokines and creating a huge reaction. It's thought that cytokine storm caused many of the deaths during the Spanish flu epidemic of 1918.
So we set up some experiment already which do not need patient samples, but we can test to see how low cytokine might regulate this response.
And when you say that it's the overreaction
Yes.
that causes damage to the tissue, is that what is causing deaths?
We don't know yet, but I think this is one of the possibilities based on the patterns in how patient progressed.
So that's the team at the Chinese Academy of Medical Sciences Oxford Institute, trying to understand the virus and our response to it better. But I'd rather not get it in the first place. What are scientists doing to protect us from COVID-19? For answers, we head over to another institute nearby.
This is Edward Jenner. Actually.
A statue of the father of vaccination stands outside the Jenner Institute. Our guide here is Sarah Gilbert, Professor of Vaccinology. She is a very busy woman, and she's not the only one. This department is on the front line of the effort to develop a coronavirus vaccine.
We're all quite sleep deprived. Been working round the clock and through the weekend. We're having to plan multiple things in parallel. So it's an exciting time. It's also a very stressful time.
Sarah and colleagues were also working on vaccines for MERS, Lassa fever and a hemorrhagic fever, all work that's been put on hold as they focus on this latest threat. They've also been working on speeding up the vaccine development process, and that would be really useful in this case. So what is it about the coronavirus that pushes it to the top of the priority list?
Well, we know it's something that is spreading very rapidly now, and we are going to need to have a vaccine against it. What everybody's now expecting is that even if it is contained, it's quite likely to come back because it's spread so widely now. And it may not be contained. It may be something that goes through the population and becomes part of the panel of viruses that causes respiratory infections every winter.
Now, the Jenner Institute, named after Edward Jenner, who basically did the first vaccine by taking cowpox and inoculating a child with it, and they didn't get smallpox. How has vaccine technology moved on since then?
It's moved on a long way, but although there are still concepts in what Edward Jenner did that we use today, he used one infectious disease to protect against another one. The vaccines that we use are based on is an adenovirus vaccine. And adenoviruses cause a cold-like disease as well. But what we've done is taken an adenovirus and removed some of its genes so that it's now very safe to use, so it doesn't give anybody a cold, to which we can add the genes from other disease-causing organisms like the coronavirus.
Now, things have moved on, though, and there's some slightly higher tech ways of approaching the vaccine. Can you tell me about how you build a vaccine?
Well, what we do is use molecular biology initially. So we have the genome of the adenovirus. We have that in the freezer, in the lab, and we've added some extra sequences to it so that we can then make more copies of it in bacteria. And then we can purify that DNA and we can manipulate it to add in a gene that encodes part of the novel coronavirus, for example, that we want to use to make the vaccine. And then we put that into mammalian cells, and that starts to form the adenovirus vector vaccine. It can only make more copies of itself in that particular cell line, so it doesn't do that when we use it as a vaccine, but that's the process that we use to increase the amount of virus that we have, and then we have to purify the virus away from the cells that we've used to produce it on. So we end up with a very highly purified preparation of the adenovirus with the novel coronavirus gene in it, and that is then essentially the vaccine.
How long does it take to make a vaccine?
In the past, it's taken anything up to 20 years from the point at which somebody decides to start making a vaccine to having something that's licensed. That means that it's ready to be used on a wide scale. We are accelerating a lot now, but the world doesn't have a good track record on making vaccines for use against outbreak diseases.
As well as a plethora of practical problems, there are ethical questions with trialling this kind of vaccine. Any good scientific trial has a control group, people who are given a dummy placebo vaccine without any potentially protective ingredient. is it fair to do that in an outbreak situation? It's a quandary that Sarah's teams and others developing vaccines around the world are currently considering. The other barrier is cost. How expensive might it be to develop this?
Well, it depends what you mean by expensive. I mean, if we compare it to what's been wiped off the stock market, it would be just an absolute bargain. Obviously, the money isn't there up front, and that's the difficulty. And the money is needed now to get these projects up and running because, if we sit around and discuss it and decide what we should do, it's too late. We started in January. As soon as the sequence of the coronavirus was released, we started to make our vaccine.
So what is the timescale? When are you going to have a vaccine developed?
It's really very difficult to say for a number of reasons. One reason is that we would normally do vaccine development in stages, and we'd complete the first stage before we moved on to the next stage. And so we know, having completed manufacturing, for example, we know we've got a vaccine in the freezer ready to do the clinical trial with. And we might run into problems with manufacturing. So all of these things have to feed into the planning together, and we're trying to work out multiple things in parallel. But we're going to face problems with possibly staff shortages, because as the outbreak spreads in the UK, we might find that people are isolated at home for a while. So that could hold us up. We're trying to do some planning around what to do about that. And it also might affect supply of the raw materials that we need. With each week, we're getting a bit closer to seeing where we might be, but at any time, something could stop us. And as the outbreak spreads, I think it's going to be increasingly difficult to do this kind of research.
So 12 to 18 months?
Those are the figures that people have been saying. We don't know. It's not going to be extremely fast. We'll have to see how things happen. To be honest, as soon as one person has tested a vaccine and shown that it works, it will help everybody else. So very, very difficult to predict.
So preventative vaccines are going to take a while. Almost certainly nothing in time to protect most people against the current outbreak if it peaks in the next few months before dying down. Before then, there might be something else to help people who are already infected. I said we'd return to Peter Horby. He's trialling drugs called antiviral treatments.
There are not many antivirals, but there are a number of existing medicines that are used for other purposes that in the laboratory, in the test tube, have an antiviral effect. And we're looking at these, and we have developed trials to try and see if they are safe, which is very important in patients, but also if they have any effect in either shortening duration of illness or decreasing the likelihood of getting complications.
And how do they work?
Some of them interfere with the binding of the virus to the cell, so it doesn't get into the cell where it replicates itself. Some of them interfere with some of the processes in the replication of the virus. So they all have different mechanisms of action. We've been working with colleagues in China since the 2nd of January on testing quite a well-used drug for HIV, actually. So it targets a particular enzyme which also is similar in the coronavirus. So we've been testing that in China to see if it has any effect.
That's quite clever. If you're testing something that's already being used for another virus, then you know its toxicity levels, and it's not like starting off with a drug that's sort of entirely cold.
Yeah, that's exactly right. There's two classes of drugs that we could try. One is what we call repurposed drugs, so drugs that are already licensed for other indications like HIV. There's also drugs that are used for high blood pressure that might work. So they're the ones that we're prioritising first to try and test. The other class of drugs is what we call the investigational drugs, ones that are not currently licensed for any illness. And they're much harder to test because you need to get a better understanding of the safety, etc.
How long does it take to find out whether one of these antiviral options might work?
The answer is, don't know. We have to get enough patients in the trials. They have to be what we call adequately powered, which is enough patients in the trial to be able to detect a difference that's clinically important. So you're usually talking, hundreds, if not thousands of patients. It can happen quickly or slowly. So at the start of the epidemic in China, we were recruiting, 20, 30 patients a day, and now it's down to, a handful each week. And then you have to wait for the patients to finish their period in the trials. So often trials will, say, assess the patient finally at 90 days after they've received the drug to see what the outcome is. We want to go a bit quicker than that. So in our trials, we've been looking at 28 days or even down to 14 days. But that still means you can't decide if a patient has recovered or not until the very last patient has reached, say, their day 28, which means you have at least a month after the last patient's in the trial before you can report the results.
Which do you reckon is going to come first, a decent antiviral or a decent vaccine?
Well, we'll have the results of the antiviral trials before we'll have the results of the vaccine trials, I'm sure. But it's, I don't hazard a guess to whether any of them will work or not. Most trials have a negative result. Most things aren't as effective as we would hope they are.
If the outcome is positive, when might people who become sick be prescribed one of those drugs?
You're probably talking another couple of months at least, I think, before you'll have a really concrete result in any of the trials that might say this drug, is effective and it could be used.
I think that's quite soon. What's the catch?
Oh, no, it is quite soon. As I said, most drug trials take years and years and years. Well, the catch is that the most likely result is that these things don't have much of an effect at all, because with viruses, most people get a mild illness and so they don't need any treatment and they don't come to hospital. The patients that come to hospital are often quite sick. And what we've seen with the coronavirus is that those patients that come to hospital are often quite late in disease and may be quite unwell, at which point treating them with an antiviral may not have a huge effect because they've already got a lot of inflammation in the lungs, etc.
So you're trying to get a sweet spot for your trials where you're trying to prove that these antivirals are effective, but there's only a point earlier on in people's infections when it can really have an impact.
Yes, and we call that the therapeutic window. It's the window where you think an antiviral can have an effect.
If you've got drugs that might work and you've got an at-risk population, why don't you just prescribe them anyway?
It's very tempting to do that, but in the long run it's counterproductive because you really need to be treating patients based on good evidence that you're giving them a drug that's both safe and has a positive effect. Otherwise, you end up in this never-ending cycle of giving the drug because you used to give it and people expect it. It's very hard to stop that practice, and it's very hard to do any future research of new drugs because you're then comparing it to an unproven standard of care.
Is this outbreak just going to be something that every country in the world has to go through?
I think it's quite likely, yes. It seems that it's very infectious and that if you put in place very stringent measures, like has been done in China, you can control the outbreak. But I think that's probably only temporary. And the expectation is that in China, once the control measures relax, which inevitably they'll have to be, because you can't stop people travelling or working, etc., then there'll be a resurgence of cases. So I think it's inevitable that every country will suffer to some extent, at least in the initial phases. It may be that in future outbreaks in future years, what we see is that this becomes a more normal, mild respiratory illness. But because it's the first time that the population's seeing this virus, we're seeing more severe disease this time around.
So it's not going to go away?
My personal view is it's not going to go away, no.
Peter mentioned the results of one of his antiviral trials are coming any day now. Head to this show's webpage at bbcworldservice.com/crowdscience, where there'll be a link to the published research once we get it. So, an antiviral drug to lessen the impact of the virus might be with us in a couple of months. From what Professor Sarah Gilbert is saying, a vaccine could be a year down the line, but really there are so many unknowns that she's unwilling to put a timescale on it. What I've taken away from visiting all of these teams is their sheer dedication to the task in hand. So, thanks, scientists. The team that brings you Crowd Science also make Health Check and Science in Action. So, if you do end up confined to quarters, keep downloading those podcasts from the BBC World Service.
That's it for this edition of Crowd Science from the BBC World Service. Today's question was from me, Evelyn, in Colorado, USA. The presenter was Marnie Chesterton, and the producer was Jen Winty. If you have a question that science might be able to answer, please send it to crowdscience@bbc.co.uk. Thanks for listening. Bye.